Dear students, welcome to one more session of Open Elective BTOE 5 Viruses, Foes and Friends. In this particular lecture, we'll try to look into the various different viruses and their classification. Classification is an age-old art of identifying the organisms based on their features. Since viruses are present in almost most of the hosts, like plant, animal, human, bacteria, fungi, as well as even algal viruses are found, we need to understand how these viruses could be classified. What are the different criteria that are used to classify these viruses so that we can identify a particular type of virus without any haste? So, classification of viruses is a very important topic that gives us insight into what kind of viruses do we encounter every day in our day-to-day life. Before we go to the classification of viruses, let us understand a bit of what exactly are the general characteristics of a virus. A virus, as I described in the last unit, is nothing but a biologically obligate parasite which consists of a nucleic acid enveloped with a protein, and this protein we call it as a capsid of capsid, and this forms a virion or a virus particle. The capsid accounts for the most of the virion mass, and it is called as the protein coat of the virus. In some of the organisms, it is very simply organized. In some organisms, it is very complex. And this particular capsid or the virus protein coat is composed of smaller subunits. It is composed of many subunits called as protomeres or capsomeres. So these protomeres aggregate together to form something called as capsomeres, which in turn repeat to form the overall capsid of the virus. In the picture, you can see there are two types of capsids that you can see here. The first capsid of the bacteriophage being in an icosahedral symmetry. And the second capsid you can see is a complex virus capsid of the influenza virus. Sometimes these capsids may also be enveloped by a lipid layer. We call that as envelope. Coming to the general characteristics of the virus, the second important component of the virion of the virus is the nucleic acid. So what kinds of nucleic acid do we have in case of most of the viruses? So the virus nucleic acids are of two types, DNA and RNA. DNA stands for deoxyribonucleic acid, RNA stands for ribonucleic acid. The classical example for a DNA virus is the pox virus, which causes smallpox. The classical example for an RNA virus is the one which caused the pandemic. We call it as SARS coronavirus, or severe acute respiratory syndrome virus. We call it as coronavirus. Now, this nucleic acid can be either single-stranded or double-stranded. Like there are viruses which have single-stranded DNA, double-stranded DNA, such as pox viruses. There are viruses which have the single-stranded DNA, for example, the sarcoviruses or the Germany viruses that infect the plants. There are certain viruses which also contain double-stranded RNA. For example, a virus known as blue tongue virus, which causes disease in the cattle. And we have viruses with single-stranded RNA, for instance, the SARS coronavirus. And these nucleic acids can be sometimes either circular molecules or linear molecules. For example, if you look into the viruses with circular dsDNA, there are different types of bacteriophage which contain circular dsDNA. There are certain viruses where there are linear DNA molecules as well as the nucleic acids. Apart from that, RNA also is of different types. Like people classify the RNA into single-stranded, double-stranded, as well as the single-stranded RNA is further classified into positive sense and the negative sense. Let me explain to you what is positive sense. When the virus particle infects a host cell, the virus releases its nucleic acid. And suppose if the nucleic acid is an RNA, and if it enters into the host cell, immediately that RNA, if it is recognized by the ribosomes, and it is synthesized into a protein or it is encoded into a protein, such type of RNA which is immediately translated is called as a positive sense RNA. In case of negative sense RNA, so this RNA of the viruses cannot be directly read by the ribosomes. Hence, they have to be further transcribed into positive sense by using the host RNA polymerase. Then further, when these are encoded into their complementary positive sense from negative sense. This positive sense acts as an mRNA to produce or to translate the proteins required for the virus. That is called as negative sense. Sometimes these nucleic acids can also be glycosylated. That is, they can be associated with carbohydrate moiety. For example, certain viruses which have glycosylated nucleic acids like influenza virus, or certain times these nucleic acids can be bound with proteins. We call them as nucleoproteins. For example, the classical example is the rabies virus nucleic acid. We call the nucleic acid of the rabies virus as nucleoprotein because it is associated with the protein. These are the different types of nucleic acids that you will come across. And further, there are certain nucleic acids which may also reverse transcribe. If you remember in the last chapter, when I was talking about the central dogma theory of molecular biology, I was talking about reverse transcription. Reverse transcription is a mechanism where the single-stranded RNA is further, reverse transcribed into a DNA, and this DNA gets integrated into the host genome and it replicates via RNA-mediated stage. For example, the classical HIV. There are even double-stranded DNA viruses which can replicate via RNA-mediated state. They also need reverse transcriptase. We can call them as double-stranded DNA virus with reverse transcriptase. So overall, if you see, there are almost 7 different classes of viruses, which we will be discussing in the further aspects of the topic. So apart from the coat protein, that is the capsid and the nucleic acid, viruses may also have something called as envelope. This envelope is amorphous structure which is composed of lipid and sometimes could be associated with some sort of a protein and carbohydrate as well. It lies outside the capsid and it is mostly antigenic in nature. This contains mosaic of different antigens of the virus. Hence, for example, if you take enveloped viruses like even SARS coronavirus has a spike protein, and most of the viruses which are enveloped can also have certain type of envelope. For example, rabies virus. Rabies virus has an envelope around its structure. So these envelopes sometimes contain spikes. For example, they can be glycoprotein projections or they can be enzymatic in action. For example, in case of influenza virus, hemagglutinin spike, which has its activity. And these spikes or the envelopes can act as the antigenic regions of the virus. They can be basically formed by a lipid membrane. In the picture, you can see this is the orange colored envelope around the Rift Valley fever virus, which you can see it's an envelope here. And this picture, you can see what I'm marking up will show you the rhabdovirus structure, which has that red colored envelope around its capsid. You can also see in the electron microscopic image of the adenovirus that there are spikes present over the adenovirus. Coming to the virus morphology. Virus morphologies are technically very complex in certain stages. Like there are icosahedral symmetries, there are helical symmetries, and there are complex virus structural symmetries. Now, what exactly is a icosahedron? Icosahedron is a type of a viral structure which can be formed based on protomeres that aggregate themselves into 5 or 6 groups to form something called as a capsomere. And the repetition of these capsomeres will give you something called as icosahedral capsids that have 12 different corners and occupied by a penton capsomere and 20 different triangular faces. This is what is a simple icosahedron. You can see in my picture, I'm showing you an icosahedron that is made up of a plain paper. Like you can see, if you are able to count the vertices, you can see 1, 2, 3, 4, 5, 6 vertices on the top and 6 vertices on the bottom, which make you 12 different corners for this icosahedron. If you observe this icosahedron from the top, I'll try to show you the topmost structure of the icosahedron. You can see that this structure is now a pentamere. You can have 5 different phases for this structure. You can see phase 1, phase 2, phase 3, 4, and 5. So it is a pentamere, or it's a what you can call a 5 faced capsomere. If we count the total number of triangular faces in this, there will be 20 different faces. This is how icosahedral symmetry can be made from two different types of shapes. One is a pentamer, and another one is a hexamer, as shown in the figure. You can see this is called as the pentamer, which I have just shown you, and there can be an hexamer as well. Hexamer is made up of 6 different facets of the protein. I'll show you a little more complex capsid so that you can understand the icosahedron. So this is icosahedron of a Zika virus. Zika virus is a virus that causes abnormalities in the pregnant woman, which leads to the abortion of the fetus. If you see into this icosahedron, this is a little more a complex icosahedron where you can see that this shape is a pentamer. You can see the five sides that is a pentagon-like shape of the different faces here. There can be even icosahedrons with hexagonal shape that we call them as hexameres as well. So there are two subunits that can basically repeat, and this leads to something called as a triangulation number. Triangulation number is nothing but the number of subunits that repeat a virus virion or a virus particle. For example, I can show you one more capsid which I made for your presentation purpose. You can see this capsid. This capsid subunit, if you can see, the subunit of this capsid is made up of 3 different colored dots. One is an orange dot, one is a red dot, and other one is a pink dot. You can see yellow, orange, and red colored dots. 3 different subunits. Let us consider these 3 dots as 3 different protein subunits that make up the virion. For instance, in this case, you can see that there are 3 proteins here: protein chain A, chain B, and chain C, which can form in this image, which can form the icosahedron shape. So icosahedral symmetry is identical a cubic symmetry, and they contain either hexons or pentons of the capsomeres. Like, how can you find a hexon? I have been showing you a penton. How can you find a hexon? Like you can see in this icosahedron, if I come to you a little near, that this shape that is showing up here, this shape is made up of a penton. And if you want to see a hexagon, if I just invert it, you can see a hexagonal shape here. This is called as a hexagonal capsomere. So I'll be giving you an assignment so that you all people can come up with your own icosahedrons that are available over internet and make your own icosahedron and submit it to me for the CA1, that is the assignment 1. There will be 2 activities for your uh, internal assessment, that is called as CA activity. So I'll be giving you to make a icosahedron for this activity. So I'll be posting you this on your WhatsApp group. But for now, we'll be looking into what exactly is this icosahedron symmetry. I hope you all understood what is an icosahedron symmetry, how it is unique. Now there is one more symmetry called as helical symmetry. And this helical symmetry is present in case of your classical example, that is the tobacco mosaic virus. It is a simple symmetry where you can observe. I'll show you on the screen. Please observe, guys. On the screen, you can see that the helical symmetry is a rod-like symmetry. And this rod-like symmetry is based on repetitions of a particular subunit. See, this is the viral capsid, you can see here, and it's a repetition in a spiral manner. It is basically in a spiral manner that each subunit is arranged to form that helical or rod-like symmetry. So these capsomeres also form a ribbon-like structure, what you can say in other terms, which will make the helix. The repetition will make the helical capsid. You can see that there is the diameter of the protomere as well as the diameter of the whole virion particle or the helical virion particle that is formed in case of the tobacco mosaic virus. So that is about the helical symmetry. Coming to the complex symmetry, like there are certain viruses like rhabdoviruses, which are having a icosahedron and a helical symmetry together. That is, if you see the virion of rhabdovirus, Or say, let us take the virion of a bacteriophage. Bacteriophage is a complex structure. How can you term this as a complex structure? You can see the head is of icosahedral symmetry in case of the bacteriophages. Then comes the neck, which has the helical symmetry, and a basal plate with the tail fibers. Hence, these viruses are said to be complex in their nature or complex in their form. So this way, that's why these viruses are called as complex symmetries. So virus morphology, if you take on, most of the viruses are icosahedral, some of them are helical, and some of them can even have a complex symmetry. Here are some of the morphologies to present to you. Like you can see the helical symmetry of the tobacco mosaic virus here, and the polyhedral structure or the icosahedral structure of the adenovirus, then a spherical structure of the influenza virus, which follows the same type of icosahedral symmetry, but it is converted into a spherical in nature in shape. And last is the complex viruses, which are the bacteriophages. So these are the some of the characteristics of the virus which we need to understand before we classify the viruses. And generally, there is a notion that people consider viruses as non-living organisms. But being virologist, I do consider them as a living organisms. They have both characteristics of a living versus a non-living organism. One good thing, one easier thing to remember is these are obligate parasites, and hence they know and they require a host to survive. And they cannot reproduce on their own. This is what people term them as a non-living, because they don't have their own reproductive mechanism. They have to depend on the host for replication. They cannot make their own energy. That means they use the host energy sources for their replication processes. They come in different shapes and sizes, usually specific for a specific host and the cell type within a specific host. These are the non-living characteristics of the viruses. If you talk about the living characteristics, most of the viruses have the capability to have a nucleic acid, a protein. They are live, they can replicate, they can form their progeny. They have some enzymes as well. These are all the living characteristics of the virus. Now, there is another notion saying that non-enveloped viruses are typically more virulent. What do you call a word as virulent? Virulency is the capability of actually forming an intense form of disease, or capable of infecting the organism so intensely that the disease can be lethal. This is called as virulency. So people consider non-enveloped viruses are more virulent than the enveloped viruses, because enveloped viruses have a sensitive envelope, and this envelope is prone to different sort of chemicals. For example, if you take SARS coronavirus, which is an enveloped virus. So in order to kill this virus or destroy this virus, we use a sanitizer. Sanitizer is an alcohol which can disturb the lipid membrane structure of the envelope, and hence the virus is inactivated. That is not in the case of the non-enveloped viruses. They are more virulent than enveloped viruses. They usually cause host cell lysis. This is a generalized theory. I don't support this theory very much, but still, since it is there, we need to understand this one. Since non-enveloped viruses also do not have a lipid membrane, cell lysis is most common mode of exiting. If it is an enveloped virus, the most common mode of exiting would be a budding. That's where when they bud out of the host cell, they take the envelope of the host cell. That's why most of these enveloped viruses attain an envelope from the host cell. So non-enveloped viruses are very resistant to extreme pH, extreme temperatures, dryness, and even disinfectant. For example, noroviruses, enteroviruses, adenoviruses, rhinoviruses. These are the viruses which are non-enveloped in nature. Enveloped viruses, they have the envelope around the capsid, and when they exist from the host cell, usually they will take the envelope from the host cell. And that's why budding is the most common way of exiting out without lysing the host cell. Enveloped viruses are more sensitive to this pH, heat, dryness, disinfectants. For example, the influenza virus, that is the flu virus, and the SARS coronavirus, they get very much easily inactivated because of the sanitizers that we use, which are alcohol based. And this alcohol ruptures the envelope, or it dissolves the envelope membrane of these viruses. This picture shows you something called as virosphere. What is a virosphere? Virosphere is a term used to describe the biosphere of the different viruses, different kinds of viruses. You could have heard the term biosphere. Biosphere is nothing but the presence of various organisms, different multiple organisms in a single ecological niche. We call it as biosphere. Earth is a form of a biosphere. Similarly, virosphere is a one ecological niche that is artificially created to understand all the different types of viruses at a single place. You can see the different classes of viruses here: double stranded DNA viruses, single stranded DNA, single stranded RNA, single stranded RNA positive sense and negative sense, then double stranded RNA, and dsDNA and single stranded RNA with reverse transcriptase. In spite of that, for example, in case of dsDNA viruses in this picture, you can observe that there are dsDNA viruses that infect bacteria, that infect fungus, that infect invertebrates, that infect vertebrates. So there are different kinds of these viruses. How do you identify these? How to classify? How many viruses can we memorize? Being human, our brain will not be able to memorize each and every virus. But still, there are ways how to understand the classification of these viruses, how to understand this virosphere itself. Let us come to the classification of viruses. In order to classify the viruses, there is a committee, we call it as ICTV. It's a committee of virology division of the International Union of Microbiological Societies, IUMS. ICTV stands for International Committee for Taxonomy of Viruses. These are the people who decide how to classify the virus. They'll give you a specific way of classifying the viruses. It was started in 1966 as the International Committee for Nomenclature of Viruses. So it was initially called as ICNV, and later it was renamed as ICTV. It is the current body which regulates how to define the classification of a virus. So this was converted into ICTV, that is International Committee for Taxonomy of Viruses, in the year 1977. Now, what are the objectives of ICTV? One is to develop internationally agreed taxonomy for all these viruses. That's what we read today. When we call SARS coronavirus to the genus coronavirus belonging to a family called as Coronaviridae, that is given by ICT. Then the second function is to establish internationally agreed names for the virus taxa, because everyone can call viruses with their disease names with different names, but there will be a unique taxonomical identification that is given for these viruses. To communicate, the next objective would be to communicate the decisions concerning the